The world was a tapestry of humid green and deep loamy brown, long before the concept of a world even existed. In the fertile, sun-drenched delta where the great river kissed the sea, two families of creatures lived in a state of wary symbiosis. In the canopy and on the ground, the arborale, slender, quick-handed and chattering, foraged for figs and tender shoots. Rooting in the soft earth below were the suidai, stout, thoughtful creatures with soft, intelligent eyes that missed nothing. The delta between them was not just ecological, but chromosomal. A quirk of primordial genetics meant their lineages, though distinct, were separated by a mere 3 chromosomes, a negligible gap in the grand, messy ledger of life. They were cousins across a forgotten divide. Their coexistence was one of habit, not design. The arborale would drop fruit, and the suidae would clean it up, their grunts of satisfaction a constant low rhythm beneath the shrieks and clicks of the monkeys. They grew to understand each other's alarm calls, the subtle language of a snapped branch or a changed wind. It began not with a cataclysm, but with curiosity. A young arborale male, whom one might call inquisitive, found himself fascinated by a suidai female, a creature of quiet depth he might have named stillness. She was not like the females of his kind. Her intelligence was not expressed in frantic leaps, but in a profound, observing calm. He offered her a mango, not dropped from a height, but placed gently before her snout. She accepted, her dark eye holding his. Their bond, forged in shared food and silent companionship, eventually culminated in a physical coupling that should have been impossible. Yet, their forms, guided by that strange chromosomal kinship, aligned. He, the male, she, the female. The act was clumsy, alien, but natural in its conclusion. Months later, stillness gave birth. The troop of arborale chattered in confusion. The sounder of suidae grunted with unease. The offspring was strange, hairless things, with the pink, soft skin of a newborn suidae, but the 5-fingered, grasping hands of an arborale. Their faces were a unique blend, a pronounced brow, but a flat, thoughtful snout. They were helpless, mewling creatures. Stillness, driven by a deep mammalian instinct, nursed them. Her rich milk gave them strength and bone. Inquisitive, for his part, supplied a constant stream of mashed fruits, nuts and soft grubs, weaning them onto a diet that combined the best of both their worlds. The young thrived. And to the astonishment of all, when they reached maturity, they did not seek partners among the pure arboralia or suidae. They bred with each other, and their offspring were fertile. This was the genesis, the first true blending. The hybrid children possessed a terrifying potential. From their arborale fathers, they inherited nimble fingers, a curiosity that demanded to take things apart, and an understanding of leverage, of cause and effect. They could not just use a stick to dig, they could sharpen it, haft a stone to its end, and conceive of it as a tool for more than digging. From their suidae mothers, they inherited a different kind of mind. It was a deep, internal self-awareness. The suidae did not just feel fear, they contemplated it. They recognized themselves as distinct entities in a vast, complicated world. They were deeply social, empathetic, and communicative in a language of nuanced grunts and gestures that spoke of inner thought. They reflected. Combined, these traits were revolutionary. The dexterous hands could now enact the will of a reflective, strategic mind. A mind that could not only see a predator and flee, but could see a predator, remember its path, and dig a pit there for the next day. A mind that could not only feel the cold, but could see the Suidae huddled together for warmth, and conceive of stitching hides together to wear that warmth. They began to manipulate their environment in a way no creature ever had. They built simple structures, not just nests. They created tools for specific imagined purposes. They pointed at the stars and grunted questions to each other, their language a pigeon of chitter and rumble, evolving into something new, something capable of conveying abstract thought. Over thousands of years, the interbreeding stabilized. The chromosomal dance settled into a new, harmonious pattern. The distinct features softened into a new form, bipedal for efficiency, with hands perfectly suited for fine manipulation, and brains vast and complex enough to hold entire worlds within them. They lost the thick hair of the ape and the pronounced snout of the pig, their features blending into a new, unique symmetry. They became more efficient, more adaptable, their mixed heritage their greatest strength. They looked out at the world their parents had simply inhabited, and they saw not just what was, but what could be. They saw a rock and thought, Amma. They saw a valley and thought, oh. They saw each other and knew, in a way no other creature did, that they were the same. In the deep, genetic memory of our species, there remains an echo of this ancient union, a faint, inexplicable affinity for the creatures of the field and forest, a remnant of dexterity and curiosity married to profound, stubborn introspection. And in time, as the ages turned and the ice retreated, this new genus, born of monkey and pig, of ingenuity and reflection, would find a name for itself. They would call themselves Homo sapiens, the knowing man, and they would inherit the earth. While the preceding narrative is largely speculative, it is grounded in scholarship and critical thinking. Consider the following scientific facts. The overall genomic DNA sequence similarity between pigs and humans is about 84%. This is high for two different orders of mammals, but significantly lower than between humans and apes. Skin and body plan. Pig skin is structurally very similar to human skin in terms of epidermal and dermal thickness, hair follicle density, and collagen content. This is why it has historically been used for skin graft research and training. Organ size and physiology. Heart. Pig heart valves are very similar in size, structure and function to human heart valves and have been used as bioprosthetic implants, xenografts, for decades. Kidneys. The size, blood filtration rate, and internal structure of pig kidneys are remarkably similar to human kidneys, making them a leading candidate for xenotransplantation. Lungs. The lobular structure and branching of the airways are similar. Pancreas. The structure and function of the endocrined pancreas are similar, driving research for diabetic treatments. Digestive system. Pigs are omnivores with a digestive system, simple stomach, longer intestine that is much more similar to humans than that of herbivores or carnivores like cats. This makes them excellent models for nutritional studies. Teeth. Both pigs and humans are heterodont, have different types of teeth, incisors, canines, premolars, molars, though the shape and size differ. Eye. The pig eye is very similar in size, structure, and physiology to the human eye, making it a common model for ophthalmological research. Many pig immune genes closely resemble human immune pathways, why pigs are used in xenotransplantation research. Pigs share many of the same genes involved in diabetes, obesity, cardiovascular disease, and Alzheimer's disease, making them useful biomedical models. Different numbers. Many regions of pig chromosomes show synteny, similar arrangement of genes in blocks with human chromosomes. Pigs have similarities in reproductive hormones and gestational physiology, though pigs have multiple offspring per pregnancy. Humans and apes are well known to share numerous characteristics. Shared primate body plan, clavicle, rotating shoulder joint, pentadactyl hands with opposable thumbs. Merely identical skull bones, jaw structure, dental formula, though apes have larger canines. Humans and apes have nearly identical arrangement of major muscles. Brain structures correspond almost one-to-one. Difference lies in size and relative expansion of the neocortex in humans. Very similar gestation length. Humans 9 months, chimps 8 months. Menstrual cycles, hormones, and reproductive anatomy are strikingly close. Humans and chimpanzees share 98.8 to 99% DNA sequence similarity, depending on how insertions, deletions are counted. Nearly all human genes have direct one-to-one homologs in apes. Differences are often in regulatory regions, gene expression timing, not the protein coding itself. In considering that our distant ancestors may have actually been the coming together of ape and pig, the similarities overall are staggering. Most everyone has heard of the ape as our evolutionary ancestor, but few are aware of how strikingly similar we are to the pig. Your average pig in a farm, trapped in a pen, has the sentient awareness of a 5-year-old human child. Human children, at that age, are among the most interesting and delightful people. The pig is never allowed to see daylight, never allowed to go outdoors, trapped in a pen and fed to death. Consider the life of that poor pig, their awareness, and our treatment of them, the next time you ask for more bacon. To look objectively and scientifically at the overlap between pig, ape, and man would require one to consider the possibility that at some point in the distant past, these two species bred an early form of human. Reproduction, not the mathematical absurdity of random mutation, was the source of our origin, as it is for all life on Earth. The very simple mathematical equation of two life forms coming together sexually to create offspring is the formula in question, and it is observable. We can observe it in our world, everywhere. It is why we are here, not from random mutation, but from sexual reproduction. That is how life works. If you like this video, please hit the subscribe button, and thank you for watching.